Welcome friends and welcome in another video tutorial of Easy Life Science. Here I am talking about antigen presentation by a MHC class first molecule, a endogenous pathway or a cytotoxic pathway. So please subscribe my channel Easy Life Science, like my video and share it to your friends. Press the bell icon so you will get a notification of newly uploaded video. So let's start. The two processing pathways are there. One is a pathway for presentation of a intracellular antigen, that is called as a endogenous antigen. The pathway for it is a cytosolic pathway, whereas another one is a pathway for presentation of exogenous antigen. For this, the pathway is a endocytic pathway. The MHC class first molecule use a cytosolic pathway for presentation of antigen. The immune system uses two different pathways to eliminate intracellular and extracellular antigens. The endogenous antigen, those generated within a cell, for example, the antigen present in a tumor cell or antigen present in a virus infected cell. These two cells shows a presence of endogenous antigen, and these are processed in a cytosolic pathway and presented on a membrane with a class first molecule. The class first molecule are expressed by nucleated cells, whereas the exogenous antigen, those taken up by endocytosis, are processed in a endocytic pathway and presented on a membrane with a class second molecule, and the class second molecule are expressed by antigen presenting cells, for example B cells, dendritic cells and the macrophages. The first, that is a cytosolic pathway or a cytotoxic pathway for endogenous antigen. And as said earlier, the endogenous antigen, the virus infected cells or tumor cells having endogenous antigens, the pathway by which the endogenous antigen are degraded for presentation with class first molecule utilizes a same pathway, the pathway which is involved in showing a normal turnover of intracellular proteins. And the MHC class first molecule shows or flag or present antigen to a CD8 plus cell, and the CD8 plus cells are also called as cytotoxic cells. First, the peptides for presentation are generated by a protease complex called as a proteasome. Here, the peptide, the meaning of peptide is a peptide antigen. The peptide antigen, that is a virus antigen or a viral proteins or a protein present in a tumor cell, are larger in size and they cannot easily loaded on a class first molecule and cannot represented or presented to a CD8 plus cell. So these larger proteins converted into a smaller fragment, and the proteasome carry out this conversion or a cleavage. The intracellular proteins or intracellular antigens are degraded into a short peptides by a cytosolic proteolytic system which is present in all cells. And for this cytosolic proteolytic system by a proteasome, the protein should be tagged with ubiquitin. The ubiquitin is one kind of protein, and this ubiquitin get attached to a lysine subunit of a viral peptide antigen or an intracellular peptide antigen, after which it is recognized by a proteasome, and the proteasome cleaves that tagged or ubiquitinized protein antigen or a intracellular antigen into a smaller fragment. And for this cleavage, by a proteasome, there is a need of ATP, so it is a ATP dependent process. In this process, a large sized peptide antigen are cleaved into a smaller sized peptide antigen, and then these smaller sized peptide antigens are ready to load on a class first molecule. After loading, the they are presented to a CD8 plus cells. Here, this is a peptide antigen. This has a larger size. It should be cleaved into a short sized peptides, for which it is tagged with a ubiquitin. The ubiquitinizing enzyme adds a ubiquitin to a lysine amino acids, and then it gets cleaved from a proteasome into a short fragments of the peptide, and these are the smaller antigens, and these smaller antigens are accessible to load on a class first molecule. Then the peptides or a peptide antigens are transported from a cytosol to a rough endoplasmic reticulum. The peptide generated in a cytosol by the action of a proteasome are translocated by a tap protein into a rough endoplasmic reticulum, and for this process there is a requirement of a hydrolysis of ATP. The tap proteins are called as a transporter proteins or a transporter associated with antigen processing. The tap protein is a heterodimer consisting of tap 1 and tap 2. These two are dimers. It is made up of three domains. One is a membrane spanning domain. Second is a domain which projects into a lumen of a rough endoplasmic reticulum, and a third domain is a ATP binding domain. It projects into a cytosol. These are the three domains of a tap protein. It is a heterodimer. The role of tap protein is that the tap protein transport the peptide antigen from cytosol to a ER. Both TAP1 and TAP2 belongs to the family of ATP binding cassette protein found in a membranes of many cells, including bacteria. This protein mediate ATP dependent transport of amino acid, sugars, ions, and peptides. And the TAP has a highest affinity for a peptide made up of 8 to 10 amino acids. And we know that the for binding on a class first molecule, there is a need of a peptide of 8 to 10 amino acids. So it is an optimal peptide length for a class first molecule binding. The tap protein transport the peptide antigen to the class first molecule which is present in a ER. The assembly of a class first molecule in a ER. In this event, as the MHC class first molecule, these are a glycoprotein or a protein, they are assembled into a lumen of a rough endoplasmic reticulum. And like other protein, the alpha chain and a beta 2 microglobulin component of a class first molecule are synthesized on a polysomes along the ER or a rough endoplasmic reticulum. The assembly of this component into a stable or a complete class first molecule with the peptide, and after loading a peptide antigen on a class first molecule, it exists from a ER. But the assembly process requires several steps. It includes a participation of molecular chaperones, and these molecular chaperones facilitate the folding of polypeptides. The first molecular chaperone involved in a class first MSC molecule assimilation is a culnexin. The culnexin is a resident membrane protein of ER, It is present in a lumen of a rough endoplasmic reticulum. The culnexin associated with a free class first alpha chain and promotes its folding. When the beta 2 microglobulin binds to a alpha chain, the culnexin is released and the class first molecule associated with the another two chaperones, cul reticulin and a tapacin. The tapacin, a tap associated protein, brings the tap transporter into a proximity with a class first molecule and allow it to acquire an antigenic peptide. As the antigen peptide loads on a class first molecule, a complex is formed, a complex of class first molecule with a peptide antigen. After this, the cul reticulin and a tapacin get released and this associated the complex MHC first molecule and a peptide antigen are transport to a Golgi complex and Golgi complex to a cell surface. An additional chaperone, that is a ERP57, has observed the role in a transportation of the complex of MHC first molecule and a peptide antigen. Here is a schematic representation of a cytosolic pathway. The virus enters into a cytoplasm of a cell and the viral antigen or a viral protein antigen, it get entered into a host cell through a vesicle, that is a phagosome. The viral antigen or peptides or these proteins are larger inside. It should be tagged with ubiquitin, and after ubiquitinization, the proteolytic degradation of that protein is there. This degradation is carried out by a proteasome, and number of shorter fragments of a peptides are formed, and these peptide antigens with the help of tap protein are loaded onto a class first MHC molecule which are synthesized in a ER. Simultaneously, the assembly of a class first molecule in a ER is going. After loading a peptide antigen on a MHC class first molecule, it is transported to a Golgi and Golgi to a endocytic vesicle and endocytic vesicle to a on a cell membrane, and the CD8 plus cell get binds through its co-receptor to the alpha 3 domain of a class first molecule, and the CD8 plus cell or a cytotoxic T cell release cytokines and granzymes, and because of these granzymes and cytokines, this infected cell or a viral infected cell get killed, and this is a cytosolic pathway. Thank you for watching my video. Please subscribe my channel, like my video and share it to your friends.